Let's move on to active transport. Now, active transport, I already gave a little introduction in the past for this. It is the transport of a substance against its electrochemical gradient across the membrane, and it utilizes energy. So you are using energy because you need to push something against its concentration gradient, meaning you're taking something from a lower concentration to a higher concentration. This could be the number of molecules or even charge that can influence, that determines whether it's active or not. So electrical and chemical gradient, you should look at both of these. Anyway, so utilizing energy, the way how these carrier mediated transports work, there are these molecules, like I said, in active transport. So there's a cell, there is a molecular protein structure, could be with multiple subunits, and molecules can bind on one side, both sides, or simultaneously over there, inside and outside, and things can flip. This molecule will flip, it will change its configuration and shape, allowing molecules to be moved across the membrane, or sometimes in the same direction, or just one molecule can be made to move in or out of the membrane. So utilizing energy could be directly where this pump or carrier molecule, which will carry out the transport, will bind to ATP directly. So an ATP molecule binds to this protein, and whatever it's about to transport will get transported, because the energy is provided by the ATP molecule directly with the high energy phosphate bond. Another way is the indirect technique over there, the indirect utilization of energy, and that's under secondary active transport. I'm going to elaborate on this some more. Just go over this classification over here and definition. Active transport is transport across electrochemical gradient, and we use energy. Now, if the transport molecule, the carrier protein, uses ATP directly, we call it a pump. So this is a pump that works over here. And these pumps have the ability to bind to certain substances and change the configuration over there. And this is through something called ping-pong mechanism. Ping-pong mechanism. Okay, ping-pong mechanism. And this uses ATP directly. So there's a site for ATP to bind to this molecule, and it will transfer its phosphate bond, causing change in the shape over here. Now, in this case, we call this primary active transport. And some examples given here. One of the most important examples is actually sodium potassium ATPase pump. This is the most important example over here, and I'll talk about this, give a small little talk about sodium potassium ATPase pump soon. Now we have Circa ATPase pump. This is found especially in your muscles, and sodium potassium ATPase pump is in all cells. Circa ATPase is in your muscles, both skeletal, cardiac, and smooth muscle. They help in the relaxing of muscles. What they do is they take calcium and pump it back into the smooth endoplasmic reticulum, which stores this calcium. You have something called hydrogen ATPase pump. This is present on your lysosomes. They are present in all the cells, and they have enzymes to digest almost every organic molecule that exists in nature. So the ability to break it down requires these enzymes to be active. And so here's your cell over here, and this cell with this nucleus has got these little granules present in them, and these granules are called lysosomes. They are like a cell inside the cell. Think of it like a lipid bilayer membrane with your enzymes present over here. These are the enzyme molecules, and these enzymes work at a certain pH. So you need the correct pH over there, the power of the hydrogen. Okay, that's the negative log of hydrogen ion concentration, and for that it needs an acidic environment, a pH of around 4. So this requires a fair amount of hydrogen to be concentrated in your lysosomes. So there is a hydrogen ATPase pump, which gets pumped inside over here. Then you have ATP binding cassette proteins. These are a family of proteins, transporter molecules. There are around 40 to 50 that are known to exist, and they transport different ions, cholesterol, lipids, ions like chloride especially, and even sodium that are transported. And if this is mutated, especially the chloride transporter, you get a condition called cystic fibrosis. The point is, all of these molecules bind to a certain molecule or ion. These transporters bind to a certain molecule or ion, and they will transport them to the other side. Sometimes they will bind to different molecules, one on one side, one on the other, and they will flip them, one this side, one that side. Another transporter could be it binds to two or more different molecules and transports them on the same side. So on the same side, when you transport more than one, it's called a symport. When you transport more than one in the opposite directions, then you call that antiport. And if you have a protein molecule, a structure which is going to take one molecule in or out, it doesn't matter, that's a uniport. It transports a single molecule over there. Next we have the secondary active transport. Now, so again, transport happens against the electrochemical gradient, and we are utilizing energy differently. We have these transporter molecules, but they don't work the way you think they work. What happens is, I'll give you an example. First of all, this is a cell, and the cell could be in your GIT, a gastrointestinal cell, or it could even be in the proximal convoluted tubule of your nephron. So in the nephron. Now what happens is, your gastrointestinal cell is supposed to absorb glucose from the lumen of the intestine and take it into the blood. So when you eat food, like you eat a little bit of a donut or something sweet like baklava, you get some glucose in your gastrointestinal tract. And that glucose gives you energy, and that energy happens when you get the glucose into the blood, and it goes all the way to the cell. So you got to get the glucose into the cell, first of all, the lining epithelium of your GIT, and even something similar happens in the proximal convoluted cell of the nephron. I'll talk to you about that. The point is, glucose has to enter into a cell, and then it has to go through that cell, and then it has to go into your blood. So how does this happen? If you look at the cell, it's already been absorbing a lot of glucose because you had a nice amount of sugar. So there's plenty of glucose in your cell already. There's plenty of glucose in your cell compared to in the lumen. So that means glucose has to enter into the cell against its concentration gradient over here. There's no talk about charge because glucose does not have a charge. So now, how is glucose going to enter into the cell? Well, there is a transporter for this, and it is called the sodium glucose link transporter, SGLT. There's SGLT1 and SGLT2. We'll just call it SGLT for now, okay? Sodium glucose link transporter. So how does this work? I'll give you an example. So one molecule of sodium is going to bind over here, and one molecule of glucose that I'm going to draw in red, because red stuff reminds you of sweet stuff. And psychologically also, if you want to make a dish appear sweeter, you put a little bit of red color. You make the plate red, and people think the thing tastes much sweeter. Okay, so this is a sodium atom, and the red color is the glucose over here. Now what happens is, the sodium on the outside of the cell, even in the intestinal tract, is quite high. You have more sodium outside the cell than in the cell. The cell on the inside has a sodium of only 14 milliequivalents. We know this from the previous lectures on homeostasis and body fluid compartments. Please watch those videos, and it will make everything easy for you. So now that means sodium, there's plenty of sodium outside, there's less sodium inside. And glucose, if we look at that, there's plenty of glucose inside and less glucose outside. Now what happens is the sodium is going to stick here and the glucose is going to stick over here to the outer surface of the cell, which is facing the lumen, meaning the hole of the pipe structure over here. And sodium, is, with its force of wanting to enter, is going to cause a conformational change in this molecule, allowing the sodium to enter, and glucose is also taken along for the ride. I usually crack a joke and tell it in a funny way. Imagine you happen to imagine it's Valentine's Day, and you want to eat at a particular restaurant, and you really want to eat, but there's a condition. Only if you bring your girlfriend will you get a 50% off in that restaurant. So it's like a couple's restaurant over there for Valentine's Day. Now imagine you are sodium, you are the boy, and you're just going there for the food. So what happens is you don't have anybody to go with at the moment. You've been too busy studying medicine, and like good kids, you've only focused on your studies. At least I hope that's the case. But you also want to have a meal once in a while. So you don't have anybody to go with. So you ask one of your cousins to just come along with you. It's purely platonic. You just pretend to be my girlfriend so I can get into the restaurant and I can eat over there. But your cousin's also kind of boring and she doesn't like to have fun, and she's scared that her friends might be in the restaurant, so there might be lots of other friends. Glucose molecules are the equivalent of your cousin. Maybe I should make that thread over here. So there's she's scared, what if they see me with you? That'll be so weird. So what happens is glucose doesn't want to go in, but sodium, because there's so much sodium outside the cell than inside the cell, it wants to go in. It drives the entry of sodium and it takes glucose along for the ride with it. So sodium is happy, it's gone inside, and glucose is concentrated in the cell. And eventually this glucose will make it into the blood. How? Because there is another transporter which is going to bind to the glucose molecule, and then the glucose will transport along its concentration gradient, because it's going from an area of high glucose to an area of relatively low glucose concentration, meaning the ECF and the blood. So there is this transporter for that, and we call it the GLUT transporter. This is like GLUT1 and GLUT2, GLUT, GLUT1, GLUT2 transporter. This is facilitated diffusion. This is using a carrier molecule, and it's just transporting it along the path. But the example of the secondary active transport is the SGLT carrier over here, the carrier molecule, secondary active transport. And this also happens in the proximal convoluted tubule nephrons over here, and it's supposed to happen this way in the PCT also. Okay, you have to reabsorb glucose, which gets filtered from the blood into the nephron, so you don't waste it out there. So the same mechanism happens also over there. And you should know about this because there are certain drugs that block the SGLT transporters, to be precise, SGLT type 2, like empagliflozin, canagliflozin, dapagliflozin. You can just add the suffix at the end over there, dapagliflozin. Now these are all drugs that block the SGLT transporters in your nephrons, so you don't absorb too much of sugar back into your blood and you urinate out the sugar. And this helps you to control your blood sugar levels in diabetes, type 2 diabetes, and also you kind of lose some weight also. It's an added benefit, and it's also been shown to protect the person from cardiac events like heart attacks and stuff. So that's another wonderful thing, and it's something you should be aware of, these drugs. So you can see how physiology is heavily influential in your pharmacology knowledge. You want to know pharmac, you have to know physiology. Then only you can become a good medical practitioner over here. So this is an example of secondary active transport, and there are many more examples out there which I will go through, and you must know about this. It is very important. So here's your SGLT, and that's going to take the glucose in, and you have GLUT over here, which takes it from the PCT into the blood vessels. The glucose enters into the blood vessels. So that is how we have the transport taking place.